So I'm going to sort of go over a little bit of what you've seen with what Nick presented, and just sort of tell you a little bit about how the lab is sort of working on seagrass and clams from the perspective of genetic lens. So the title of my talk is From Stress to Strength, and so the focus here is to try to, instead of identifying the things that are killing everything and the stress, why things are stressed, and I sort of want to flip the coin and look at the other side and ask, what are the mechanisms that organisms can use to deal with that stress? So our research here in Florida Oceanographic and our research program is focused on a growing paradigm shift that focuses less on understanding how stressors induce negative effects to one that's focused more on understanding the mechanisms through which organisms and ecosystems are resilient to stressors. So we ask questions like, what makes organisms resilient to stressors? And we ask, can we leverage that information to advance restoration? And we try to incorporate genetic or evolutionary rescue into our restoration strategies. So there's a difference between genetic rescue and evolutionary rescue, and these have been used, for example, in the California condor. They focused on genetic rescue, which is to restore the health of a shrinking population by adding new genes to fix inbreeding damage. There's another concept, evolutionary rescue, and this is something that naturally happens to protect populations experiencing environmental stress from extinction through natural selection. So there are literally populations of elephants now that don't grow tusks anymore because they survive because they're not killed by their top predator, the human. So we all have probably heard about genetic bottlenecks, and this is when a population goes through this sort of random situation that culls certain alleles from the population. Certain genes are no longer found due to random death of the population, where genetic diversity is lost. So genetic bottlenecks cause reductions in genetic diversity, and we know from countless experiments and much work that this reduces the evolutionary potential of species. But is increasing diversity always the answer? And so when this genetic bottleneck is caused by a stressor, then we need to consider the resilient genotypes that are coming through this sort of stress filter. An accidental or intentional introduction of maladaptive genotypes, so these ones that should have been selected against, can cause outbreeding depression. So we have this idea of inbreeding, but we need to think about outbreeding as well. And so these blue guys, which are less adapted, can slow or reverse this evolutionary rescue. And so we can think about this graphically if we notice that abundance here of our population is on the y-axis, and we have this environmental stress that begins, and that causes this abundance to decrease, so the population is decreasing, and genetic diversity is also decreasing. But these adapted alleles or adapted genes can become more numerous over time, and this is called a selective sweep. When beneficial genes or alleles go to fixation, they become more abundant in the population. So if we are just throwing these organisms out there during our restoration, and we're doing the best we can, we're trying to put animals and plants back out into the environment, but if we're doing that randomly, then we could be adding alleles and genes that are maladapted, and we can cause problems and basically disrupt this natural process of evolutionary rescue. So what my lab, what our research program is focused on is trying to understand if we can use modern genetics to understand what evolutionary mechanisms drive resilience to stressors, and whether or not we can leverage that to balance inbreeding and outbreeding depression during restoration. So we're using these ideas and investigating these concepts within the Indian River Lagoon, which you've been introduced to by my team already. We know that the Indian River Lagoon suffered a stress-induced regime shift, which means that it was a seagrass-dominated environment, and it shifted to an algae-dominated environment, and this started happening in 2011. And there were mass die-off of seagrass and bivalves during this time. So before I got here, my team was investigating what the effects of adding clams during seagrass restoration are, and whether or not those effects would be stronger if they protected that grass and those clams from herbivores using fences. And so they did this 2x2 experiment where they crossed clams and no clams with fences and no fences. The seagrass that they used came from the meadow just right outside. And so they collected the seagrass from here and they planted it at these two sites, Haines and FOS. Haines is just south of Fort Pierce and FOS is right outside. We could walk there. And the clams came from Todd Osborne, and he collected clams that survived this 2011 mass mortality event, and he refers to these clams as super clams because they were the ones that were out there surviving. It took his team 8 weeks to find about 37 clams. That's how bad the mortality event was. Unfortunately, right after I got here, and I swear it wasn't my fault, there was a mass mortality event at the seagrass sites. And so I got here and the team was like, let's go see the seagrass. And they went out and took me out there, and basically most of the seagrass was dead. And many of the clams died as well. And so what we decided to do was, instead of crying and being sad about the situation, which everybody was devastated, we decided to flip the coin, and we collected the survivors of the grass and the clams and compared them back to their original populations. So our grass's original population was this meadow, and Todd has his brood stock that we could compare him to. And so we were interested in, what was different about those creatures that actually survived. You've seen this before. So this is a phylogeny of our grass outside. So I'm starting with the grass data. And what you see in green are the individuals that we collected from the meadow. Haines is in red, and these are survivors from that Fort Pierce site, and the survivors from our site at FOS are in blue. And so the first thing I want to draw your attention to is the fact that I should tell you, this phylogeny allows us to understand who is most closely related to who. So these two individuals are very close together, and their next most closely related individuals are these. And we go down. So, M7 is not terribly closely related to this this grass that survived behind our facility. So these are meadow individuals that we did not find survivors for. And then this individual is a survivor from FOS. These are survivors from Haines and FOS and so on. What's really cool about this is that we know that, for example, M18 that we collected in the meadow is very close closely related to this survivor as is M30. So we went out to our meadow and we collected those individuals, and we're growing them now in the front of house seagrass nursery in our genetic library. We also went out and collected some of these individuals that are not related to the survivors so that we can compare. What else do I want to tell you? So the next thing we looked at with this data is an admixture model. Admixture tells us about the ancestry and describes how organisms are interbreeding. The K value describes the number of separate genetic groups, and here we found that 2 groups are the most appropriate. And so each of these bars is an individual that we can identify, or that we sampled. And the length of the color represents the percentage of DNA that aligns with that specific ancestral cluster. So these individuals over here are all from one ancestral cluster, and these are from a different ancestral cluster. And then we have these sort of different amounts of each group. And one thing that we see is that these individuals from the meadow that do not have representatives that we found surviving in the field are mostly part of this blue ancestral group. And then the individuals that are survivors are really clustered in this area that has more and more and more of this red. So that gives us some indication that these populations are different, and that we have different genes that might be controlling this ability to survive these stressors in the IRL. So next, we are intentionally propagating the strains that were more resilient, as well as ones that we have no evidence that they were resilient because they did not have survivor representatives. And we're developing a living library gene bank so that we can go back and investigate the genetic structure of those specific individuals that are related to survivors or not. We're also gearing up to start testing each survivor to determine what environmental stressors they're resilient to. And we're identifying the genes that were selected. And I'd like to build a gene array, which means that once we know what genes are different between survivors and non-survivors, we can set up basically a way to sample populations and say, do you have this resilience gene? Do you have this resilience gene? And so we can do this in the wild, so we can go throughout the IRL and look at the populations. It's called seascape genomics. And look at which populations have different resilience genes. And we can also go around to our other nurseries and do this in our nursery to ask, what resilience genes do we have on property? What resilience gene does HBOI have in their nursery? Now, I told you we had clams out there too, and we sampled clams and did a similar investigation of their genetic structure. Our hypothesis was that these super clams, they do have names. I think my team is probably getting so sick of it, but it was so funny to me. So our hypothesis was that these environmental perturbations basically were equivalent to large-scale natural selection events, leaving the hardiest, most stress-resilient individuals. So the idea was to test through sequencing their genome whether or not they were different from other populations. So we started off by looking at Sapelo Island clams, Matanzas River clams, and these super clams that Todd Osborne has in his lab. But then we added, after this mortality event, we added our Haynes survivors and our FOS survivors so that we could understand sort of what they look like relative to these other populations. So this is another phylogeny again. These two individuals are more closely related to each other than they are to this guy. And what we see is that there are two major large So this Sapelo Island in red and Matanzas in blue form one group. And the IRL superclans, which are these ones that survived FOS and Haynes, and then the ones from Todd's lab are in purple, form this other group, starting here and going here. So they're different. The IRL clams appear to be different from the clams from Sapelo Island and Matanzas River. Now we can look at K again, and it's a little bit unclear. It's our separate groups. The data suggests that this could be separated into 2 or 3 groups. And so I have 2 groups here and 3 groups here. So if we look at the PCA, this allows us to summarize multiple aspects of the data and put the data into groups based on similarity. And so what we see is that you can't even tell the difference between Matanzas and Georgia. They're all clustered up here. They're very similar. And when we look at the IRL and FOS and Haynes, these are all produced by Todd in his lab through aquaculture. And what you notice is they sort of focus on or fall out into these 2 separate groups, which helps us decide that maybe the interpretation of 3 populations is the best one. And so, so this ancestry plot shows that Matanzas and Georgia are most closely related to each other. They sort of come from the same ancestral grouping. This IRL and FOS, so IRL, remember, are just the clams that Todd had in his lab, and FOS are the ones that survived out in the wild, and Haynes are the ones that survived. So these that survived at Haynes look very different. We also looked at nucleotide diversity, and it turns out that these Haynes clams also have a higher diversity. What's really interesting is if you look at Tajima's D, this allows us to understand population contractions or expansions. And so a D that is below 0, or sorry, above 0, usually happens when there's a sudden population contraction. And so it totally makes sense that Todd's clams from his lab have undergone this population contraction. Remember, he only found like 37 clams or something like that. And then our FOS clams look very similar. But what's interesting is he's been able, through breeding, to make this population that has a lower D value, which signifies a selective sweep, so that something interesting has happened, a population expansion following a bottleneck. And so this has been artificially created by Todd, kind of by random. And now that we know that we can do this, we can do it on purpose. This also shows that the FST value shows how distantly related these organisms are, and Haynes comes out as very different. And remember, this is all from that same family. And so it shows that you can really sort of manipulate which genes are in which organisms, or which alleles, I should say. So our next steps for this really are to sequence the broodstock, but we can't crack them open to do that because that would kill them. And so what we've thought about doing is sequencing their germ cells. So as Michaela pointed out earlier, these are broadcast spawners, and so you can trick them into spawning, and that's what clam farmers do this all the time. And so we can trick them into breeding, and then we can collect their germ cells, and then we can sequence those germ cells to figure out who is most related to those ones that survived. And a variety of other questions can be answered. Then we can do selective crossing to make animals that we think are going to be very resilient or not. And we can then heat waves or for water quality and do stress tests, just like in the grass. And then we would conduct genetic analyses on the survivors and the non-survivors, so that we can test for differences in their genomes. And what I'm interested in ultimately is understanding which genes are associated with survival. And that way, again, we can develop a PCR array to test for tolerance genes in the same way that I described for the grass. Then, when we have all of this, we can start to design breeding programs for intentional restoration. So once the clam farmers know that they have these resilient populations, then we can be more intentional with our restoration. So ultimately, our department here, focusing on research at FOS, what we really want to do is identify these mechanisms that promote survival, rather than focusing on all the reasons why things are dying. And we feel like this is the approach, a better approach, in order to understand this global change and develop innovative mitigation strategies. With that, I have a ton of people to thank. My entire group has helped with this, discussing this, going out in the field. I just put up a spattering of people who have sort of helped more hands-on with this work. And I always like to thank my super grass and super